to record this for my A-Day, y'all. So let's see. And y'all are like y'all did great on the last recording. Y'all were good. All right. So Mendelian genetics. Who was Gregor Mendel? Yeah, he's the father of genetics. You should be able to tell me, Gregor Mendel, he was the father of genetics. You don't have to write any of this. I mean, you should have most of it in your journals, but have your notes open because I might want you to write a few things. Remember, he used pea plants to determine genetic probability. He was very important to the study of genetics. And you got to admit, man he had a clean haircut to be a monk. All right. That was fly. All right, Johann. Let's see if I can skip down to what's really important. Okay. Terminology. Now, this I already went over last week, so this should be old news. So, genotype. Whenever we're looking at a genotype, you're looking for letters. You're looking for letters that represent a gene. Now, you might see this word on the test. Don't be afraid. Does anybody have the definition to that word? What is that? What's an allele? What's an allele?
Two forms of a gene.
Thank you. It's a different form of a gene. So let's just say we were talking about cats. And I say cats, they have two, these particular breed of cats have two coat colors, white and brown. What would be the allele in that situation? Yeah, the white and brown, a different variation of coat color, white and brown. Now the phenotype, what is a phenotype? It's a physical feature. You can see a phenotype. Can you always see somebody's genotype? No. For example, you have couples who have a baby, and both of these people have brown eyes, and the baby comes out with blue or green eyes. That's because these couples carry an allele that is recessive. So you don't always see recessive alleles. Let me give you an example. All right, let's just say for eye color, we say brown eyes is big B. So that individual is purebred brown eyes. They have brown eyes, and the only gametes they're going to make are going to have brown eyes. Now let's just say an individual has this. Okay, yeah, just conveniently don't work for me. Thanks. All right, what color eyes do they have? What's their phenotype? Brown. What makes their eyes brown? The dominant gene, the big B. What is their genotype? Starts with an H. Heterozygous. This one is homozygous dominant. This one is heterozygous. It's hetero because they're different, but the dominant gene takes over. So I just want y'all to know there's a difference between genotype and phenotype. Okay, let's see. I think y'all are ready for practice problems. All right. And actually, a lot of my students, y'all complain about the recording being too long, even though we take notes for like 60 minutes every other day. But you know, the customer's always right, right? Right? No, that's a lie. That's not true. That's completely false. Customer's mostly wrong. We just need them. All right, here we go. This is a worksheet you're gonna get. I haven't given you the worksheet yet because I want you to do the first practice problem in your journal, not on the worksheet. And then you're going to retrace those steps on the worksheet. So on your paper, I want you guys to do this practice problem with me. If you've watched the recording to this far, if you're one of my students watching, boom, you get answers 1 and 2 for free. Let your peers struggle and probably fail. All right, so it says right here, monohybrid cross practice, give peas a chance. For each problem, use the Punnett square to show your work. Now, these are the steps that we're going to do for each one of these problems. Step A, show the parents' genotypes as a cross in the form of, you know, this genotype crossed with this genotype. Provide a Punnett square, just can't read this morning. Provide a Punnett square to predict the outcome of the cross. List the possible genotypes and the percentage. List the possible phenotypes and the percentages. Now, these are universal steps that I want you all to follow. before we even do this. So write how to solve a monohybrid cross. So write this somewhere on there. So how, nope. So let me give you the steps. What does monohybrid mean? One single trait, right? So here are the 4 steps. Step 1, you make a key. Step 2, find parent genotypes. Two more steps. Step 3, plug into the Punnett square. And then lastly, step 4, find genotypic and phenotypic ratios. So those are your 3 steps to solving a monohybrid cross. 3 steps. I think Punnett square has 2 T's. My bad. All right, everybody got the steps? Oh, okay. So now is at the point of your education in biology where you should be able to link all things. You should be able to link. You should be able to go back to the very first lesson that I taught you guys about this stuff. The well, not exactly this, but remember when I talked about evolution and mutations and all these other things, how animals have this survival instinct that forces some animals to go extinct, and how animals change over time. This all has to do with passing down successful traits. When you take your test in like a month and a half, you're going to be expected to bridge all of this stuff. So I've been teaching you in pieces, but now is the time where you should be able to take your journals, flip all the way back to the first 9 weeks, and draw some similarities between everything. Those are the students that master the test. All right, so let's go ahead and do the first 2 problems together. I'm pretty sure like the majority of my students have stopped watching the recording, so you're going to get these first 2 for free. All right, in pea plants, purple flowers are dominant over white flowers, which are recessive. Cross 2 homozygous dominant PP parents. So you're crossing 2 homozygous dominant individuals. What's our first step? Paula, what's my first step? What do I do first? Make a key. So everyone, let's make a key. So we're doing number 1 together. So here's our key. And I want y'all to do this every time you solve a monohybrid cross. Our key is going to talk about our alleles and our phenotypes. So our 2 phenotypes, we have purple, and then you have white. Now when you have to attribute a letter for these traits. I always go with the dominant. So if purple is dominant, and then I'm going to do a lowercase p. Now what you can do so you don't confuse yourself is you can draw a line under your dominant one so you know that's the dominant trait, and because these might get confusing. All right, so now I have step 1 done. What's step 2? Anthony, what's step 2? All right, so parent genotype. Now this one is easy, and it's telling us to write the genotypes like this, so I'll write it like that. So your mom, and then you're crossing it with dad. And actually, it'll be something like this because you have 2 alleles for each trait. But anyways. All right, what 2 genes does mom have? if you're crossing two homozygous dominant parents, what two genes does mom have? Big P. You gotta remember it's capitalized. Big P. And dad also has two dominant P's. So this is gonna be real easy. You're crossing two purebred parents. Trenton, what's step 3? All right, so you're gonna plug it into the Punnett square. So right now y'all are doing these easy Punnett squares. The hard Punnett squares are where you have 16 boxes. You only have 4 right now, and we will do 1 or 2 with 16 boxes. All right, so that's my Punnett square, and then I put the genes. Big P, Big P, Big P, Big P, and then you bring this one over, this one down for every single box. So since this one is purebred, you're only going to end up with this. The offspring will have all the dominant traits of the parent. So it says list the possible genotypes and phenotypes and the percentages. So this is how you would do this part. So this is where you're going to get your answer from. So I'm going to move over here. So I'm going to write a G for genotypes. You only have one genotype. That's Big P, Big P. And the fraction is 4 out of 4 or 100%. Now the phenotype, I'll put P for phenotype. You're going to say purple. I don't know why it's changing like that, yo. Purple. You're going to say 4 out of 4 or 100%. That's how you solve that. So that's easy whenever you're crossing that. So on the STAR test, they're not going to give you one that easy. But that's just a demonstration so you can start using that. And if you look at number 2, I do want y'all to go through the steps when you get your worksheet, but I'm not going to do that. I'm going to cross a different one. I'm going to cross number 3 because these become like the most difficult ones sometimes. So we'll do number 3. If you got number 1, number 2 is not hard at all. You're just crossing, you're doing the same thing but with recessive. And remember, if you have 2 recessive genes, you're going to display the recessive trait. All right, and I'll show you what I mean in a second. So let's look at number 3. All right, it says NP plants, yellow seeds are dominant to green seeds, and green seeds are recessive. Cross 2 heterozygous individuals. Gerardo, can you give me my first step? Make a key. Does anybody know how I would make my key? What 2 traits do I have? Green and yellow. Okay, which one is dominant? So we can use a big Y. Well, let me make an obvious big Y. You know, writing was never my strong suit, to be honest. And a little y. Okay? So we have big Y and little y. So we know our dominant one. I made it worse trying to fix it. Let me fix this up. There we go. All right. Now, we're crossing 2 parent. Well, I'm getting ahead of myself. What's step 2? Thank you. We need to find the parent genotype. So we know the mom has 2 genes. And she's going to be crossed with the dad, who also has 2 genes, because they have 2 genes for every trait. So the mom's genes are going to be what? Big Y, little y. And the dad, same thing. You're crossing 2 heterozygotes. Okay? Now, this is when we talked about this process called meiosis. Does anybody remember what meiosis is? No? Yes, what is it? Ryan, pay attention. What's meiosis? Meiosis. Yeah. Meiosis is the process of making reproductive cells. We talked about it when we talked about how plants get fertilized. The male part of the plant is going to go through meiosis to make pollen, which is the sex cell. The female is going to go through it to make ovules, which is the female reproductive cell. So in this situation here, this mom plant is going to have to divide her genes in half. That's called meiosis. That's where sex cells come from. So the mom is going to create eggs, and the dad is going to create sperm cells. So here we go. Here's my Punnett square. And remember, the egg and the sperm come together and they make a new individual. That's why we all look different, even from our brothers and sisters, because we have different combinations of genes coming together. All right, so the mom's gametes. What's a gamete? What's a gamete? You need to know that a gamete is a reproductive cell. A gamete is an egg or a sperm cell. So the mom's gametes or her egg cells are going to be big Y and what? And the dad's sperm cells are going to be what? What? Big Y, little y. All right, and then you cross. You bring one over, big Y, and then you bring this one down, big Y, and then you bring this one over. Always put the capital first, or you're gonna. I remember making a dumb mistake on my biology test when I was in high school. I mixed these up. I put the lowercase first and the big one Y, and then it just confused me. Always put the capital first. So in this box, now you're gonna move this one over here, big Y. Move this one down here, little y. And then in this box over here, you got this one coming over here, this one coming down here, so little y, little y. Here's the grand finale. We need to get our ratios. So I'm writing a G. What does that G stand for? Genotype. So here's our genotype ratio. How many different genotypes do we have? 3. Very good. We have big Y. We have big Y, and then we have little y. And here are our percentages and ratios. How many out of there are homozygous dominant? Big Y. 25%. What's the fraction of that? 1/4. Very good. Or 25%. How many are heterozygotes or heterozygous? 50. Very good. Or 2 out of 4. And then our last one, 1 out of 4 or 25%. Final steps, P. What does that P stand for? Come on, y'all. Stay with me. What does the P stand for? Phenotype. What are the 2 phenotypes? Green and yellow. All right. How many will be green? Nope. Oh, wait. Yeah. Who said 25? Yeah. Only he'll be green. How many will be yellow? 75. So this one will be yellow. These 2 will be yellow. Now you remember when I said earlier that you can carry the recessive gene and still have the dominant trait. So so how many will be green? We say the green will be 1 out of 4 or 25%. And then lastly, our yellow will be 3 out of 4 or 75%. That's how you solve these problems. Seems like a lot of work, but wait till y'all do dihybrid crosses. Any questions on this? Everybody get it? Kinda? You'll get it with practice. All right, let me see if I have anything to add.